Today we will discuss the sequencing genes and genome, and I will introduce you all the technique that have been used for the DNA sequencing and also the latest development in the DNA sequencing. And for the DNA sequencing, the fundamental principle is the DNA synthesis. So as we remember during the DNA replication, that's the DNA synthesis. So for DNA synthesis, you need the DNA polymerase, and then you need the deoxynucleotide, right? DNTP, and then you perform the DNA synthesis. And based on this principle, then the very first technique that developed is called chain termination sequencing, which is the Sanger sequencing. This is the most important sequencing technology because this technology been used for many years, and even nowadays that has been developed into using the same principle, and then develop into one of the next generation sequencing technique. And then after the chain termination sequencing, and later on, and the scientists developed the parallel sequencing, and the parallel sequencing also later on developed into the massive parallel sequencing, which is also one of the NGS technology. So in the beginning, we will start from the chain termination sequencing. So for the chain termination sequencing, the idea is about these two different nucleotides, all right? So this one is a DNTP, all right? And this one is dideoxynucleotide. And the difference is the 3' here, 3' here, here we have the hydroxy group, and here instead of hydroxy, it's only hydrogen. So basically, remember when you perform the DNA synthesis, it is very important that the 3' hydroxy group will attack the alpha phosphate of the incoming nucleotide, all right? And then so they will release the pyrophosphate and then form the phosphodiester bond, all right? That's from this oxygen. And now if you remove the oxygen, then the DNA synthesis will be terminated. So this one we call dideoxy-NTP, and this one the deoxy-NTP. And because this one will be no longer functional, so that means you cannot add any nucleotide after this nucleotide. So that's why it's called chain termination sequencing. So that means every time when you add the nucleotide with the dideoxynucleotide, the reaction will stop, okay? And so basically, the principle is you prepare the identical single-stranded DNA molecule, and then you anneal a short oligonucleotide to the same position on each molecule, and then you synthesize the new DNA strand, and at the place where they incorporate the dideoxynucleotide, then the synthesis will be terminated, and then you will see the different lengths of the DNA molecule. After the geolithophoresis, then you will see a series of the band, all right? That's the idea. And so basically, the more detail be like this, okay? When you perform the DNA synthesis, and then you mix the regular DNTP, all right, with one particular dideoxy NTP. So they say in this diagram here, so they say we perform the DNA sequencing. So basically you have a single-stranded DNA template, and then you have a primer, right? And then you mix the primer with the template. So you have a thousands copy, millions copy of the primer and the template in the reaction tube. And then you mix these together in the presence of DNA polymerase, and also 4 different types of the DNTP. So you have a DATP, DCTP, DGTP, and the DTTP. Plus, in this particular experiment, we add a dideoxy ATP, all right? Dideoxy ATP. So that means when you mix everything together and by chance, wherever you need to incorporate the DATP, it could be the dideoxy ATP was incorporated, all right? So for, let's say for this molecule, when they perform the synthesis, then they synthesize the second strand all the way until here, and then by chance they add the dideoxy ATP at this position. Then the reaction stop, okay? And then for this DNA molecule, so they are synthesize a new strand until this position. Then they incorporate the dideoxy ATP by chance. So and then for this DNA molecule, then they synthesize until here, then they stop. And then for this molecule, they are synthesized until this second T and then stop because they incorporate the dideoxy ATP. And then for these three DNA molecule, then you generate these three different lengths of the fragment. All right, 3 different lengths of fragment, and then you perform the gel electrophoresis. Okay, and of course you have to perform 4 different reactions. This is the first reaction. You put the dideoxy ATP. Then you can have a second reaction. You only add the dideoxy GTP, and then third reaction you add the dideoxy CTP, and fourth reaction you add dideoxy TTP. So you need to perform 4 different reactions. Each reaction you add one specific dideoxy NTP. All right. So after this reaction, then you perform the gel electrophoresis. So that for this length, all right, for this length now be the reaction you add the dideoxy ATP, and for this length, all right, the fragment here, that's the result, you add the dideoxy CTP. Okay, and then for this one dideoxy GTP, and this one dideoxy TTP, so that you see this fragment. Each of these fragments represent the position of that particular nucleotide. So for this fragment that represent at this position is A, all right, this position is C, and this position is G, and this position is T. All right, so from this gel electrophoresis, then you can read the sequencing, and from the bottom of the gel, which is the shortest fragment, that means the shortest fragment will be the one that closest to the primer, and so that you know the primer sequence, so that you can start reading from the bottom and then continue to the top. So when you read the sequence, that will be C, G, A, T, T, G, A, T, C, A, G, T. So this is the very early day. You read the sequence manually. All right, that's correct. You have to perform the chain termination sequencing reaction, and then you run the gel electrophoresis, and then you have to read it manually. That is correct. You have to read it manually. Okay, in the early days, like until 1995. Okay, 1996, around in the 90s until late 90s, you still need to read this manually, and then later on, because the development of the technology, so later on they are, instead of using this technique, because this you use the radioactive isotope, all right, radioactive isotope, so that you'll be able to expose those activity to the X-ray film, and then you can read the sequencing. And later on they develop the technology. Okay, so basically they incorporate the fluorescence dye, okay, fluorescence to each individual dideoxy nucleotide. All right, so then that means after you perform the reaction, then each dideoxynucleotide give you different color of the fluorescence, and then they give you different lengths. All right, and then they sort these fragment based on their length, and then they run this geolithophoresis, okay, and then go through a machine with the imaging system that can detect the fluorescence, and so they can read the color. All right, so they can read the color. The machine can read the color and produce this report, and based on the color, then you know the sequences. So for example, the green that'll be the C, and the red will be G, orange is A, and purple is T. From this peak that give you the fluorescence the machine detect, and then they give you this profile, and the profile the computer converted to the sequence automatically. All right, so this is the technique they developed in the late 90s, so that you use the fluorescence and plus the geolithophoresis, then you can read the sequence automatically. Okay, so that's also here. So you have capillary gel because they saw the fragment based on the size, and they could run through this gel, and then the computer, the laser detector, they detect the fluorescence and then send the signal information to the computer, and then generate a profile and give you the sequence data. So this is the chain termination sequence. It's also called Sanger sequencing because the person who developed this technology, his last name is Sanger. Okay. And then for the DNA synthesis during the sequencing reaction, so basically you need a DNA polymerase. Okay, DNA polymerase, and so basically use the clinol polymerase, okay, which does not have the 5' to 3' XOR nuclease activity. And then also have another one commonly used is called a T7 sequenase. This one is commonly used in the early days, right? And then the single-stranded DNA template, because perform the sequence, the chain termination sequencing, you need this template. So the template basically, you have to prepare the template. So to perform the sequencing, then you need to do the gene cloning first. So the gene cloning, then you can produce the double-stranded plasmid DNA, and then you have to convert the double-stranded DNA to the single-stranded DNA, because you can denature the double-stranded DNA through the boiling or alkaline denaturation, and then you can perform the sequencing. And another method that to produce the single-stranded DNA template is to clone your target DNA into the phagme, like a M13 vector. And when we discuss M13 vector, M13 particularly specifically can produce the single-stranded template, single-stranded DNA. So for in early days to perform the chain termination sequencing, and all the researchers, they most likely they all clone their target's DNA into the M13 vector, so that they can produce a single-stranded DNA and for the DNA sequencing. Okay, so that's a single-stranded DNA template. And for the primer, so for the to perform the sequencing, and if you have many DNA molecule that are required for the DNA sequencing, then normally you use the universal primer. So universal primer, so that means the universal primer is that can come from the vector sequence, not the insert. Remember, you insert the DNA molecule. That's your target DNA molecule for sequencing. But and then you can clone into if you have 20 different DNA molecule for sequencing, then you can clone into all 20 different molecule can clone into the same vector, so that you can use the same primer, because they all come from the same vector. So you can use the same primer. So that's the universal primer, so that you can sequence 20 different DNA molecule, use the same primer. Okay, so that's the universal primer. Okay, if you design a primer at the inside the inserted DNA, that will become the internal primer. But those internal primer will based on the sequence that you go in, the DNA molecule that you use. And for the DNA sequencing, okay, normally you will need to do you need to perform several times just to confirm the DNA sequence at a particular position. So for example, in this example, so in this region, so they perform 6 times, perform the sequencing 6 times. So each time we call one read. So for 6 read, so that means you repeat that same region 6 times. Okay, so that's what this means, read. Okay, each read, I mean you sequence 1 time, and then you sequence several times, so you have several read, and then you can confirm the sequence at the particular location. So for example, in here, the read 1 give you C at this location, and the read 3 give you C, but read 4 give you T, and then C, and then from these 5 different read, then you can confirm this position will be C. Okay, so that's what this means. And especially for those NGS, we also try to do a many read, so that we can confirm the sequence. And then for the pyrosequencing, okay, that's the second technology, pyrosequencing. So pyrosequencing based on the fact that when the hydroxy group of the 3' hydroxy group attack the alpha phosphate and release the pyrophosphate. Okay, and those pyrophosphate can then turn into the ATP and then provide energy to perform the enzyme reaction. Okay, and then the enzyme reaction then will produce the chemiluminescence, and so that the machine will detect the chemiluminescence and then determine that particular nucleotide is added to the sequence. And that's the idea, that's the principle for the pyrosequencing. Okay, so for this the cartoon here, so basically you have template, then if you add a DATP, if it's not A, then this would be degraded. If it's not T, it will be degraded. If it's G, all right, then if they incorporate the G, then they will release the pyrophosphate, and then this pyrophosphate, okay, will turn into the ATP, provide energy for the enzyme activity and to produce the chemiluminescence signal. Then the computer will record the signal, so that they record the G is added, and then for the next run, and then they continue to add these deoxynucleotide one at a time, and then to record a signal continuously. So that's called pyrosequencing, and this is the principle. So basically, this nucleotide attack the alpha phosphate of the incoming nucleotide, and then remove the pyrophosphate. Then this pyrophosphate, all right, can then turn into the ATP, all right, in the presence of ATP sulfurase, and then they turn into the ATP, right? And then this ATP provides the energy to perform this enzyme reaction in the presence of luciferase turn the luciferin into the oxyluciferin, and then release the light, so that the detector will be able to detect the luminofluorescence, and then they know the nucleotide is added to the sequence. All right, so that's a pyrosequencing. So any questions so far for the chain termination sequencing and the pyrosequencing? Very good. So, and then after these two technology been developed and then been used in the research in the industry for many years, and later on slowly they've been thinking like early 2000, right? So, and like late 90 and early 2000, when they tried to sequence the human genome from the Human Genome Project, they realized this is very slow. If we want to sequence in the whole human genome, that would take because the whole Human Genome Project started 1993. At that time, in the beginning, they expect to finish the whole human project in like 20 years, 15, 20 years. And then, so then the science community start thinking this is very slow process to sequence the genome. So people start thinking in what way that we can, instead of one at a time, and how can we do several at a time, right? Like 20, 100, 1,000 at the same time. And that's the beginning of the next generation sequencing, NGS. And also because of this Human Genome Project in the late 90s and early 2000, and people start thinking how to develop the sequencing technology to simultaneously sequence hundreds at a time, and that's why we develop this NGS, and also we complete the human genome draft in the early 2000s, finish the whole human genome sequencing. Okay, and so the development of the NGS, okay, as I say, early 2000. So we have these four different, in general, these four different technology, right? So we have massively parallel pyrosequencing, all right? So that's a modification from the pyrosequencing. And then we have a sequencing by synthesis. And this is the modification of the chain termination sequencing, and which is also a very popular one. And we have also a sequencing by ligation. And this is the only technology that you don't need to use the DNA polymerase. Okay, so for the pyrosequencing, sequencing by synthesis, these all need the DNA polymerase to perform the DNA synthesis to determine the DNA sequence. But for the sequencing by ligation, you do not need the DNA polymerase. You only need the ligase to perform the DNA sequencing. And also, another one is the sequencing by semiconductor technology. And that's also based on that we will discuss later about the semiconductor technology. So let's discuss the first one, the massively parallel pyrosequencing. So basically, in this massively parallel pyrosequencing, so you collect the for example here, break the genomic DNA into the fragment, all right, and into like 300 to 500 base pair long, and then each fragment, okay, you ligate to a pair of adapter, and one adapter at the either end, to the either end of the sequence. So you basically take these the DNA fragment mixed with the adapter and the present ligase, then they will ligate the adapter to the both end of the sequence. So this adapter basically they can enable a DNA fragment to be attached to a small metallic bead, because we want to attach this fragment to the bead, all right? And then also the beads here. So we have this biotin, and then we have this streptavidin biotin attachment. So we want this attached to here. And then the second role of the adapter is you can provide a annealing site for a primer for the PCR. Okay, because you're going to perform a PCR reaction to amplify this template, all right, in an oil emulsion. And then each of these, so basically you have adapter help the fragment attach to the bead, and the beads has the biotin and the streptavidin biotin attachment, and then also the adapter can serve as the annealing site for the primer, and then they can start synthesize this and PCR reaction. So you would perform a PCR reaction in the oil emulsion, all right? And of course, each of these drops will be a deposit into each of these wells, all right? So they can perform the synthesis simultaneously, okay? And then, so after PCR, then each of these beads transfer to the well, and then you can perform the parallel sequencing so that you can read it. So they have the genome, and then you sonicate the DNA, right? You use a sonicator, and then to sonicate the DNA, so it become like the small fragment, all right? And then attached to the beads, because the adapter, the DNA fragment with the adapter attached to the beads, all right? And it's on the slide, and then this is the beads. You have these biotin, streptavidin biotin attachment, all right? And then beads in water droplet, all right, in oil emulsion, and then perform the PCR, all right? Perform the PCR, and then you can you after you perform the PCR, then you distribute into the wells, all right, and into the wells, all right, and then the DNA on the beads was be subject to the parallel sequencing, all right. So the polymerase bind, all right, and then they perform the parallel sequencing. So every time they add one deoxynucleotide, if it does not, and then in like 96 wells, so you have one nucleotide, you are added to the plate, and then the computer, they will detect the signal in which well, and then after that they wash and then add the next nucleotide, and then the computer continue to record the signal from the well, and so they just repeat these 4 nucleotides, and then they perform like 150 to 200 cycles, all right? Each cycle you do 4 different nucleotides one at a time, and they perform like 100 to 200, okay? And so that the computer can record the signal from each well for the 150 cycle. So normally, the massively parallel sequencing can do up to 150 base pair in a single experiment. The advantage of parallel sequencing is that it can be automated in a massively parallel manner, so that we can do hundreds of thousands of sequences at the same time, all right? So that's the parallel sequencing. And then the sequencing using the reversible chain terminator. So the chain terminator has basically the nucleotide has been engineered just like this, all right? So now remember they use the idea about a chain termination sequencing. So the 3' hydroxy group, instead of removing the oxygen from the 3', they put a blocking group here, all right? And in the meantime, the nitrogenous base being attached to the fluorescence dye, okay? For the the difference between this one pyrosequencing, each of the 4 nucleotides must be added sequentially in separate cycle, okay? And then for this one, you can add all 4 at the same time, and only one will be added, or one will be attached to the growing strand, because only one is complementary to the sequence, all right? And so only one is added, and then once that one is added, the reaction stop, and then the computer record the fluorescence, because each of these give you different signal. So like the GTP give you a pink color, and the G is yellow, and oh sorry, these should be different. AGCT should be different nucleotide, and then each one give you different fluorescence signal. So you add all 4 at the same time, and then you wash away, only one stay here, and then you record a signal. After you record the signal, then you cleave the blocking group and also cleave the fluorescence dye, and then you perform the next cycle. And then the next cycle, you add all 4 again, only one will be attached, and then you record the signal. So for the sequencing by synthesis, it's a reversible chain terminator, and you add all 4 at the same time. For pyrosequencing, you have to add one at a time, all right, to perform the sequencing. All right. So remember, for the reversible chain terminator, the nucleotide has been engineered. So we have this 3' blocking group, all right, and also we have this the fluorescence dye attached to the nitrogenous base, and each nucleotide give you a different fluorescence dye. So this is the sequencing by synthesis, all right? And now, because this technology is very important, so we are going to watch a video about this technology, okay? Oh, by the way, this technology mainly is used by Illumina, all right, the Illumina's company. And also I will discuss this later in the end of the lecture about Illumina.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing, and data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional...
Remember, all the template, you need to put the adapter, okay?
Motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos.
Index, and also these complementary to the oligo on the later on they will show very important. And index is for you to identify which fragment you are doing. That computer will give you the index, and so that later on you'll be able to read your own sequence.
Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with Elan, composed of two types of oligos. Hybridization is enabled by the first of the two types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands.
Okay, the adapter will bind to this oligo that already attached to the cluster, right? Okay, very pay attention to here, right?
A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters.
So this will be just like a PCR reaction to amplify the same sequence here, right?
Resulting in clonal amplification of all the fragments.
And then if you have like a million different fragments, then they can also do this cluster at the same time as well.
After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands.
So you have forward strand, all right? So these all, let's say you have like a 10 different sequence here, and then 10 different sequences has been amplified, all right?
The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain.
Because they add all 4 at the same time, so that's why they compete to add it into the growing chain, right? Only the complementary one will be attached.
Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away.
First read, I mean you read one time, right? First read.
In this step, the index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away. This entire process generates millions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments. Genomic data can be securely transferred, stored, analyzed, and shared in base space sequence.
All right, so that's the general process for the sequencing by synthesis, right, which is very important and very popular technique nowadays, right? And then another technique is called a sequencing by ligation. And sequencing by ligation is the only technique that does not need to use the DNA polymerase, okay? And they just use the ligase to perform the DNA sequencing. So basically, to perform this sequencing, all right. So basically, you need to have the oligonucleotide, and it's the oligonucleotide are 8 nucleotides in length, which is octamers, and then those 8 nucleotides, you have 2 non-nucleotides at the 3' end, which is query, and then any nucleotide in the next 3, which is degenerate, right? And then you also have a sequence that is common, universal to all of the oligonucleotide at the 5' end. And then you should have a set of 16 different oligonucleotides representing all possible combination of 2 nucleotides in the 3' query end. So you can have a AA, AC, AG, AT, CA, CC, CG, CT, and TA, TC, TG, TT, GA, GC, GT, GG. So 16 combinations. So you need to have 16 different oligonucleotides, all right? And so that'll be these 16 different nucleotides, all right? So you have this, the first 2, that's a 3' end, all right, query sequence. These are different, and then these are all the same. And then each of these will carry a different fluorescence signal. Okay, they have different signal. And then basically in the beginning, you have the adapter. You should have adapter, okay, joined to the 5' end of DNA template that to be a sequencing. So you should have adapter, because the adapter can serve as the binding site for a primer, all right? So so primer bind to the adapter sequence and then provide a 5' phosphate end for ligation to the 3' hydroxy end of the adjacent hybridized octamer. And then sequential cycle of the octamer ligation extend the complementary strand, and each cycle enable determination of the query dinucleotide sequence. So one ligation cycle consists of the different steps. Like step 1, you have pools of different octamer, each tagged with a different fluorescence dye, are added along with the T4 DNA ligase, and the reaction conditions are set so that the ligation can occur only if the base at the 3' end of the one octamer are complementary to the template. And then after washing away of the non-ligated octamer and also other component, the fluorescence signal is recorded, and the identity of the dinucleotide in the query position is determined by the fluorescence signal. And then the fluorescence dye is removed after each cycle by cleaving the terminal universal nucleotide. And then the cleavage provide a free end of ligation for another octamer in the next ligation cycle. So in this figure, just show you that the situation that you have the adapter near to the template, and then primer near to the adapter. Then you mix these with those 16 octamer, and only one, because these 2 sequences CC, so only the one that carry the GG will bind, all right? One the GG bind, then they will record a signal. After record a signal, they cleave these universal sequence at here, and then they advance to the next round. And the next cycle again, they perform these ligation again. So only one that with the GT will bind, all right? So So they just follow these, repeat this cycle until the end of the sequence, so that you can see they can determine every 2 nucleotide, all right, and 2 nucleotide, and then you have a 3 nucleotide gap. So it determine 2, and then 3 nucleotide gap, and then 2, all right. And once you finish this one, then you move to the second reaction. The second reaction, basically, you have a different primer. So the first primer, you will match this from CG, CC, AT, ATAC, AG. And then for the second round, all right, so this is primer number 1. For the second round, then you move the primer, all right, there's 1 nucleotide away from the original previous primer. So originally you have this TATC, and now you move 1 to the left, so it's only TAT, and then you add 1 extra nucleotide here. So that means the primer 2 will give 1 space here, that move 1 nucleotide, all right, to the left. And so now you can determine every 2 nucleotide, and then skip 3 and 2 nucleotide. So now you, in primer 1, you determine these 2. In the primer 2, you determine these 2, all right? And then the primer 3, you shift 1 nucleotide, then you determine these 2. And then primer 4, another 2, and primer 5. So you need 5 different primer so that you can cover all the sequence. So you just you perform the 5 reaction and repeat the cycle, and then you can determine the sequence. So that's the sequencing by ligation. Okay. And then for the sequencing by the semiconductor technology, basically, this one is based on the effect that if we go back to the sequence principle here. So from here, if you look at the panel A here, so basically the hydroxy 3' hydroxide attach this alpha phosphate, then not only they release the pyrophosphate, and look, originally you have the hydroxy group, but after the forming of the phosphodiester bond, this hydrogen also is released. Okay, this hydrogen is released. So this one is released to the environment. Therefore, the pH will drop because the hydrogen ion concentration increase. So the pH will drop temporarily. So that based on this principle, then this technology, all right, is they develop this technology to determine, to measure the change of the pH, so that they know which nucleotide is added to the sequence, and then determine the gene sequence. Okay, so that's the sequencing by semiconductor technology. And then of course now they develop the 3rd generation sequencing using the nanotechnology. And for this technology, they just force the DNA sequence go through the channel, and then they can read it directly, so that you don't need to perform the amplification, synthesis, or the fluorescence detection. Okay, so this is the direct sequencing. That's the 3rd generation sequencing, but still it's not the quality is not stable, so it's not still not popular yet. Okay, and so for now, still the NGS, next generation sequencing, is the most powerful and most commonly used technology, especially in the like a year ago when the pandemic started, and it's very powerful, so that scientists can use the NGS technology to determine the SARS-CoV-2 virus genome sequence. Because after you determine the sequence, then you can develop the technology to perform the PCR reaction, the QRT PCR reaction, to determine the the presence of the virus in the human body. Okay, and also based on the genome sequence, and that's how the Pfizer and Moderna, they developed this messenger RNA vaccine. Okay, so that's why the DNA sequencing is very important. So now I just finished the overview of all the NGS technology, and here just to give you the summary of the NGS technology. Okay.
Next-generation sequencing, or NGS, is a powerful platform that has enabled the sequencing of thousands to millions of DNA molecules simultaneously. This powerful tool is revolutionizing fields such as personalized medicine, genetic diseases, and clinical diagnostics by offering a high-throughput option with the capability to sequence multiple individuals at the same time. Sanger sequencing, first developed in the 1900s, is the gold standard for DNA sequencing, and it is still used today extensively for routine sequencing applications and to validate NGS data. It utilizes a high-fidelity DNA-dependent polymerase to generate a complementary copy to a single-stranded DNA template. In each reaction, a single primer, complementary to the template, initiates a DNA synthesis reaction from the 3' end. Deoxynucleotides, or simply nucleotides, are added one after the other in a template-dependent manner. Each reaction also contains a mixture of 4 dideoxynucleotides, one for each DNA base. These dideoxynucleotides resemble the DNA monomers enough to allow incorporation into the growing strand. However, they differ from natural deoxynucleotides in 2 ways. 1, they lack a 3' hydroxyl group, which is required for further DNA extension, resulting in chain termination once incorporated in the DNA molecule. And 2, each dideoxynucleotide has a unique fluorescent dye attached to it, allowing for automatic detection of the DNA sequence. As a result, many copies of different length DNA fragments are generated in each reaction, terminated at all of the nucleotide positions of the template molecule by one of the dideoxynucleotides. The reaction mixtures are loaded on the sequencing machine, either manually onto slab gels or automatically with capillaries, and are electrophoresed to separate the DNA molecules by size. The DNA sequence is read through the fluorescent emission of the dideoxynucleotide as it flows through the gel. Modern-day Sanger sequencing instruments use capillary-based automated electrophoresis, which typically analyzes 8 to 96 sequencing reactions simultaneously. Next-generation sequencing systems have been introduced in the past decade that allow for massively parallel sequencing reactions. These systems are capable of analyzing millions or even billions of sequencing reactions at the same time. Although different machines have been developed with various differing technical details, they all share some common features. 1. Sample preparation. All next-generation sequencing platforms require a library obtained either by amplification or ligation with custom adapter sequences. 2. Sequencing machines. Each library fragment is amplified on a solid surface with covalently attached DNA linkers that hybridize the library adapters. This amplification creates clusters of DNA, each originating from a single library fragment. Each cluster will act as an individual sequencing reaction. And 3. Data output. Each machine provides the raw data at the end of the sequencing run. This raw data is a collection of DNA sequences that were generated at each cluster. The differences between the different next-generation sequencing platforms lie mainly in the technical details of the sequencing reaction and can be categorized in 4 groups: pyrosequencing, sequencing by synthesis, sequencing by ligation, and ion semiconductor sequencing. In pyrosequencing, the sequencing reaction is monitored through the release of a pyrophosphate during each nucleotide incorporation. The released pyrophosphate is used in a series of chemical reactions resulting in the generation of light. Light emission is detected by a camera, which records the appropriate sequence of the cluster. The sequencing proceeds by incubating one base at a time, measuring the light emission, if any, degrading the unincorporated bases, and then the addition of another base. This technology is capable of generating large read lengths, much comparable to the read length of Sanger sequencing. However, high reagent cost and high error rate over strings of 6 or more homopolymers have reduced its applications,
For more details on the technical aspect of this technology, please visit our knowledge base at the link provided in the description below. Sequencing by synthesis utilizes the step-by-step incorporation of reversibly fluorescent and terminated nucleotides for DNA sequencing and is used by the Illumina NGS platforms. All 4 nucleotides are added to the sequencing chip at the same time, and after nucleotide incorporation, the remaining DNA bases are washed away. The fluorescent signal is read at each cluster and recorded. Both the fluorescent molecule and the terminator group are then cleaved and washed away. This process is repeated until the sequencing reaction is complete. This system is able to overcome the disadvantages of the pyrosequencing system by only incorporating a single nucleotide at a time. However, as the sequencing reaction proceeds, the error rate of the machine also increases. This is due to incomplete removal of the fluorescent signal, which leads to higher background noise levels. Our NGS, an introduction knowledge base, provides more technical details about this technology. Sequencing by ligation is different from the other 2 methods since it does not utilize a DNA polymerase to incorporate nucleotides. Instead, it relies on 16 octamer oligonucleotide probes, each with one of 4 fluorescent dyes attached to its 5' end, but are ligated to one another. Each octamer consists of 2 probe-specific bases and 6 degenerate bases. The sequencing reaction commences by binding of the primer to the adapter sequence and then hybridization of the appropriate probe. This hybridization of the probe is guided by the 2 probe-specific bases and upon annealing is ligated to the primer sequence through a DNA ligase. Unbound oligonucleotides are washed away, then the signal is detected and recorded. After that, the fluorescent signal, along with the last 3 bases of the octamer probe, are cleaved, and then the next cycle commences. After approximately 7 cycles of ligation, the DNA strand is denatured and another sequencing primer, offset by 1 base from the previous primer, is used to repeat these steps. In total, 5 sequencing primers are used. The major disadvantage of this technology is the very short sequencing rates generated. Ion semiconductor sequencing utilizes the release of hydrogen ions during the sequencing reaction to detect the sequence of a cluster. Each cluster is located directly above a semiconductor transistor, which is capable of detecting changes in the pH of the solution. During nucleotide incorporation, a single hydrogen ion is released into the solution and it is detected by the semiconductor. The sequencing reaction itself proceeds similarly to pyrosequencing, but at a fraction of the cost. Please view our knowledge base for further details on ion semiconductor sequencing and the sequencing by ligation techniques.
So that's the summary of the DNA sequencing. So now you learn all the sequencing technology. Any question? Very good. So the next topic is how to sequence a genome, right? How to sequence a genome. So to sequence a genome, there are 2 different approaches. Okay, one is called a shotgun approach, right? And the other one is called a clone contig approach. So basically, for the shotgun strategy, basically you have to create a shotgun library. And to create a shotgun library, then you need to produce. You collect the genomic DNA. You break that genomic DNA into the fragment, right? Into fragment, and then you insert this fragment into a universal vector, so that you can generate a sequencing template, right? To obtain a random overlapping fragment, the genomic DNA is usually sheared, and the shearing conditions are optimized to obtain the uniformly sized DNA fragment. Okay. And then after you form those the vector, you clone those this fragment into the universal vector. So the resulting recombinant DNA molecule, we call it a library. And this library we introduce into the E. coli separately for amplification, and then also use it as the sequencing template. And then during the sequence, we have the because universal prime vector, so we can use the universal primer, okay? And then to perform the DNA sequencing. After we perform the sequencing for all the fragments, then the next step for the shotgun approach is to identify overlap between all the individual sequences, okay, that are generated through the sequencing reaction. Okay, the identification process must be accurate and unambiguous so that the correct genome sequence is obtained. And the shotgun sequencing has been successful with many bacterial genome. And that's because these genomes are small. And just like the COVID-19 genome, anyone know how big is the SARS-CoV-2 genome? Anyone know the SARS-CoV-2? Yes, I know it's small, so that's why I'm asking. Anyone know the size of the COVID-19 virus genome? Oh, this is the most important genome that you should know that affect the whole world and that affect the life, affect everyone's life. Is about 30. Yes, that's right. It's about 30 kilobase long, all right? It's about 30 kilobase long for SARS-CoV-2. And then the clone counting approach, okay? The clone counting approach involves a pre-sequencing phase so that you know the sequence and and then you have the overlap in the clone, and then you identify, and those are contiguous series. We call it a counting, and each piece of a clone DNA then can be a sequence. All right, and the clone counting approach can provide more accurate sequence than the shotgun approach. In a large genome that contains repetitive DNA, okay? But for the clone counting, it's time consuming, all right? Because you need to construct the overlap series of a cloned DNA fragment. And so it's more time consuming. So this is just to show you the to identify the overlapping sequence so that they can so that you can place the sequence together, all right, so that you know which one next to which one. The problem with the shotgun sequence is that possibly the incorrect overlap is met between the two sequence reads that both end within the repeat sequence, so that lead to the loss of a segment of the DNA in between the two sequences, right? So it's just like this sequence 1 and sequence 2, and because they overlap, however, they are from the identical or repeated sequence. But you don't know that, and accidentally you might put these two together and then to think this is the sequence, all right? So that's the possible problem, and this can be resolved by using the clone counting approach. And this is just to show you the in the early days when they try to first sequence the bacteria, right? The Haemophilus influenza. So they have collected genomic DNA, sonic K into fragment, and then perform the electrophoresis, and then they collect the DNA at least region 300, the 1 to 2 kb long, and then clone into the vector, and then you prepare the genomic library, and then you sequence each one, and then use a computer to generate the genome. And this is the chromosome walking. So basically, this is a technique used to assemble clone contig. Okay, so the chromosome walking is basically you have different contig, 1, 2, 3, right? And then you have different the probe at each end. They give you 1, 2, 3, 4, 5, 6, and then you have a genomic library. Then from this probe, okay, from this probe, so they say you use the oligo probe 2, all right, and then hybridize the whole genome library, and then you see the signal at this location. So you know this signal contain the probe number 2, all right, probe number 2, and then you use the whole other probe to hybridize with the genomic library, and then you see the probe number 5 also give you the signal at the same location of the number 2. Then you know that the contig 1 and contig 3 must be next to each other, all right? And then after this, then you can continue. So that's called chromosome walking until you identify all the contig. So that's called contig assembly by the chromosome walking. And also when you assemble this sequence, normally you need to have a reference genome so that you can correctly identify and to place those read in the correct place. Okay. And the one problem arise when you use a reference genome, it could be just give you the same the sequence, but in different location in the chromosome. All right. So that could be in different region. So that could be the problem cause. And when you have these different rearrangement, and because of due to the recombination, and then you might just accidentally put these 2 genes in the wrong location. Okay, that's the possible problem. And this we just discussed to give you incorrect position, because if they are the repeat sequence and the sequence 1, sequence 2, since they overlap, but you can mistakenly put these 2 together and just miss this the sequence here, all right? And the first eukaryotic chromosome to be sequenced, that's the Saccharomyces cerevisiae chromosome number 3, right? So they use the COSME, and then to clone all the DNA fragment from chromosome 3, and then from the phagome, then they sequence each of the sequence, and then assemble the sequence. That's the chromosome number 3. And this is a chromosome walking. So basically, this is more detailed than the previous one. So basically, they say you have this different genomic library, all right? You have number, and then you use the DNA from the clone A1, hybridize the whole genomic library, and then you see the signal at the B4, I5, I4, B4. So you know A1, B4, I4 are next to each other, all right? So you can have I1 in the middle, I4, B4 are next to the A1, all right? And then you can continue. You use I4. I4 is the probe to probe the genomic library, and then you see I4, and then you find A1 and F2, so that you know A1 and then F2, and then for the next probe you can use F2. Then you can identify another another clone, all right? And then they say clone A5. And then from here, A5, you use A5 to probe the whole genomic library and to identify the next one. So this is called a chromosome walking, okay? And the rapid method for clone counting assembly. So you identify the pair of the overlapping clone, all right? So basically each pair you find identify the overlapping. And so you see the F2G6, and then you see F2I4. So you know the F2G6I4 will be together, all right? And then you can find the next one, I4, F2, I4, A1. So I4 next to the F2 and connect to A1, I4, F2, and then A1. And then A1, I4, B4. So the next one will be B4. And then B4, A1, H7. So the next one is H7. So this is rapid method of clone counting assembly, just to find the overlapping pair. And so that it will be quickly to determine the arrangement of the counting, the clone counting on the genome. So the simplest approach, digest each clone with one or more restriction enzyme and look for pairs of a clone share the restriction fragment of the same size after the gelatophoresis. Okay, so that's how you determine. And the repetitive DNA PCR interspersed repeat element PCR, i.e. PCR, which is a type of PCR use primer digest designed to anneal within the repetitive DNA sequence, all right? And direct the amplification of the DNA between the adjacent repeat, all right? So you have the primer, and then you perform the PCR, and then perform the DNA electrophoresis. And then if you see these the same size fragment, so that means the clone 2, clone 3 overlap, all right? So that's how you identify the overlap pair. And the sequencing, the assembly by the sequence tag site content analysis, that's STS, all right? So you just search for pairs of clone contain a specific DNA sequence that occur just at one location. And we call that sequence is the sequence tag site, STS, all right? So just like this, so you have collection of clone and try to find a specific sequence, and then try to see any identified sequence in another clone. If you find the 2 and 4, they share that specific sequence, so that we know those two must overlap. And that sequence we call the sequence tag site, STS. Okay. And the sequence STS particularly important for the clone counting assembly, because these STS within the genome will have been determined by the genetic mapping or physical mapping. And so the STS, we can use that to anchor the clone counting onto the genome map. Okay, so that's STS. And the SMP, that's a single nucleotide polymorphism, just like this, one single nucleotide difference. That's SMP. And RFLP, the restriction fragment length polymorphism, are special type of SMP. Okay, so that'll be result in the restriction site change. So just like this, one nucleotide change, and then the restriction site disappear. And then after PCR and the restriction digestion, then you can see the difference. That's RFLP. Okay, and then the short tandem repeat, also called a microsatellite. They are made up of this repetitive sequence of 1 through 13 nucleotide in length. Okay, the length head to tail, the number repeat presenting particular STR varies, usually 5 and 20, between 5 and 20. Okay, that's STR. So just like here, STR in here, you have dinucleotide, CA, 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 and then you have 3 repeat, and you have 4 repeat. So after the PCR reaction, you perform the gel electrophoresis, then you will be able to see the difference and then determine the short tandem repeat. Okay, so that's the difference. That's TR. And the physical mapping with a mapping reagent. So that's the collection of the overlapping DNA fragment spanning through the chromosome genome that's being studied. And the mapping reagent can be the clone library, right? Or can be a radiation hybrid, okay? So that's the for the physical map. And then to use the mapping reagent, okay? So they can deduce that whether the marker would be close to each other or not. So for example, in here, number 1 and number 2, all right, they appear in 4 different fragments. So that means these 2 are very close to each other. And then for number 3 and number 4, for example, they only share 1 fragment, okay? And so that means they are further away. They have the relative far apart from each other. Okay, so this is the way to determine. Okay, to use the mapping reagent. And the physical map is generated by the directly located position of a specific sequence on chromosomal molecule. Okay. And then it can be the DNA marker that's express a sequence tag, which is a short sequence obtained from the end of a complementary DNA, the cDNA. And EST is partial of the gene sequence. And then when used in the map construction, they provide a quick way of locating the position of a gene. Okay, that's EST. And also, yesterday we talked about FISH, can also use to determine the location of particular gene. And maybe perhaps you have heard of this, that the CSI, that we received the funding to set up this genomic research facility, because we applied the funding that we received, like $1.25 million, so that we're going to acquire the equipment to set up the genomic research facility. So we will acquire those NGS sequence, and this is the design that in the 6S. Did you know 6S138, the lecture room? And next to the lecture room, there's a bigger room, and so we're going to set up that room as they convert that room into this NGS research laboratory. Okay, so it's very advanced. That we will acquire all the advanced equipment, and so that would be used for the research and also for the classes in the future. So originally the plan is we're going to open this research facility July this year, but because of the pandemic, it has been delayed since March last year. So right now we don't have any progress. So that means we will delay 1 year, because last year, right before the college closed, shut down in March, we are already have a list of the equipment and to purchase, and also we also have the architect to design the renovation of the room next to the 6S138. And however, and then because of pandemic, the college shut down, and so the whole project has been paused. So now we have to wait for the college to reopen so we can restart the whole process. So hopefully we should be able to have this facility open in spring next year. Okay, and so this is the plan for the next few years for our genomic facility. Yeah, especially for students, they can benefit for this facility because they can really learn this NGS technology. And as you can see, for this pandemic, it's so important that you can collect the sample and purify the genomic, either the DNA or RNA, and then perform the DNA sequencing. And once you determine the DNA sequence, then you can design the test, so that you can design the test to determine whether the person infected with the COVID-19 or not, or use the sequence to design the vaccine. And so in the future, so that's why it's very powerful, and that's the reason why we get the money, and then we're going to set up this facility. Okay, so that'll be it for today's lecture.